Hello everyone and welcome to today's session. And today we'll be discussing the NCERT Exemplar questions for the chapter Principle of Inheritance and Variation. So this is the part 1 of the lecture. So in this lecture we'll be discussing some of the questions and rest of the questions we will discuss in the next session. Before starting the lecture, I just want to give you one information that the images, text and other materials used in the presentations are for education purposes only. We respect the intellectual property rights of the original creators and owners. If you are the owner and have any concerns or issues, please contact us at sathee@itk.ac.in and we shall remove the content immediately. Please note that parts of the content may have been generated using generative AI, which is experimental. So today we'll be understanding the basic concepts of the chapter Principle of Inheritance and Variations by working through NCERT Exemplar questions. Okay, so today we'll be discussing example questions for the chapter Principle of Inheritance and Variation. Okay, so there are multiple choice questions, very short answer type questions, and long answer type questions. In total, we'll be discussing 25 questions today. Let's start with the first question. All genes located on same chromosome. Okay, they are. So what are the options? So these are multiple choice questions that we are discussing. What are the options? Form different groups depending upon their relative distance. Do they form different groups? Option number B, they form one linkage group. Option number C, will not form any linkage group. And option number D, form interactive groups that affects the phenotype. So what will be the correct answer here? So let's have a look at the answer for this question. So they will form one linkage group. Now let's have a look at the explanation. Now all the genes present on a particular chromosome form one linkage group. Clear? Remember this. All the genes which are present on a chromosome, this forms one linkage group. Now the number of linkage group of species correspond to the total number of different chromosomes of that species. Okay. And it is not simply the number of chromosomes in the haploid set. So there is a difference between number of chromosomes in the haploid sets and the total number of different chromosomes. So there is a difference. Okay, for example, in human males you have 22 pair of autosomes and males have one X chromosome and one Y chromosome. So actually there are 24 different chromosomes. Okay, 22 autosomes, one X chromosome, one Y chromosomes. In males, you will have 24 linkage groups. Whereas in females, you have 22 pair of autosomes and a pair of X chromosome. So in females, you have 23 linkage groups. Okay, so number of linkage groups of species correspond to total number of different chromosomes and not the number of chromosomes in haploid sets. In humans, you have 46 chromosomes, means 23 pair of chromosomes. So 23 is the haploid number. But actually, you can see in males, you have 24 linkage groups. Next question, conditions of karyotype 2n plus minus 1 and 2n plus minus 2 are called. So you can see there is a gain and loss of chromosome happening here. Gain of one chromosome or loss of one chromosome, and in 2n plus 1 there is a gain of two chromosome and loss of two chromosomes. Options are aneuploidy, polyploidy, allopolyploidy or monosomy. So what will be the correct answer here? Okay, the correct answer here will be, let's have a look at the answer. Aneuploidy is the correct answer here. So what is aneuploidy? Aneuploidy is a gain or loss of the chromosomes. Clear? So organism can gain or lose one or more chromosomes, but not the complete set of chromosomes. They only lose one or two chromosomes, or they gain one or two chromosomes. What is polyploidy? So polyploidy is actually the gain of an entire set of chromosomes. So if the cell was N, it will become 2N. Okay. So it can be triploidy, means 3 set of chromosomes, tetraploidy, 4 set of chromosomes, or pentaploidy, which means 5 set of chromosomes. Okay, what is allopolyploidy? There was one option, allopolyploidy. So it is the polyploidy in which chromosome sets are non-homologous. Okay, allo means other. Okay, allopolyploidy is derived from a stock which is heterozygous. Then there was one term, monosomy. What is monosomy? The process in which one chromosome is removed from diploid set of chromosomes. So when there is a loss of only one chromosome, this is known as monosomy. Next question, you have distance between the genes and percentage of recombination shows. Clear? Does it show a direct relationship, an inverse relationship, a parallel relationship, or there is no relationship? So what will be the answer here? They're asking about the relationship between the distance between the genes. Suppose there is a two gene A and B. What is the relationship between the distance between the gene and the percentage recombination? Okay, so here let's have a look at the answer now. Direct relationship is the correct answer. So, crossing over separate the genes away from each other. Now the distance between the genes and the percentage of recombination shows an direct relationship. Means more the distance between the genes, more will be the percentage recombination, or vice versa. Okay, so when genes are closely related, when they are placed close to each other, when the distance between the two genes is less, then they will be showing high linkage and they will exhibit low recombination frequencies, or percentage of recombination will be less. Clear? So you can say distance between the genes is directly proportional to percentage recombination. The next question, if a genetic disease is transferred from a phenotypically normal but carrier female. So female is a carrier. So females are usually carrier because they have one set of X chromosomes, one pair of X chromosomes. So carrier female is actually normal. So there is a genetic disease which is being transferred from a carrier female to only some of the male progeny. So this disease is, what the option of autosomal dominant, autosomal recessive, sex-linked dominant, or sex-linked recessive? What will be the correct answer here? So let's have a look at the answer now. Okay, it is sex-linked recessive disorder. Okay, so most sex-linked conditions, they are recessive. Okay, for X-linked diseases, the female must be homozygous for the disease allele as they possess 2 copies of the X chromosome. Okay, when will the female exhibit that disease? When she consists of X chromosome and on both of these X chromosome, mutated gene is present. But this is quite rare. Clear? Now, male must be heterozygous for a disease allele as they possess only 1 copy of the X chromosome. So males are X and Y. So even if only one mutated gene is present on the X chromosome, the males are affected because there is no counter normal allele on the Y chromosome. Now, a female with a mutation in 1 copy of a gene on 1 chromosome is carrier. So females are usually carrier. So if they have one mutated gene on X chromosome, they have normal gene present on the other X chromosome. Okay, so you can see here, this is unaffected carrier mother. Okay, this is being crossed with unaffected father. Clear? Then you can see, okay, in the progeny, all these combination will be formed. The male will transfer Y chromosome to the sons, and males can transfer X chromosome to the daughters. Now this carrier mother, she will transfer X, she can transfer what? The normal X chromosome also in the offerings, and she can transmit the X chromosome which has mutation also. So there is a 50% chance. Clear? So they can be unaffected carrier daughters, unaffected daughters, affected sons, and unaffected sons as well. Clear? So you can see all these, you can say, phenotypes can be formed here. Okay, so what you have to do here, female is a carrier, means female is heterozygous. On one of the X chromosome there is mutation, on another X chromosome there is a normal gene. Father is normal. Okay. Now for X-linked recessive disorders, we know that the carrier mother who has a mutation in gene on the X chromosome. Okay, only on the one X chromosome. Carrier mother can transmit either the X chromosome, which have this mutation, or she can transmit the X chromosome, which is having the normal gene. Okay, clear? Now there was one more option, autosomal dominant inheritance. It refers to the pattern of inheritance of a condition directly due to dominant faulty gene located on the autosome. So here, the mutated gene, and this mutated gene is dominant actually. Okay, here the mutation is dominant. And this gene is located on the autosome. That's why it is known as autosomal dominant inheritance. What is autosomal recessive? Here the mutation which occurs in the gene, this mutation is recessive and this gene is present on the autosomes. Clear? Then sex-linked dominant. So it is a very rare trait that is caused by a single abnormal gene which is present on the X chromosome. And here also, these genes are present on the X chromosomes. That's why they are sex-linked. And the mutation which is happening on the X chromosome, this is dominant. Next question, in sickle cell anemia, glutamic acid is replaced by valine. Which are on the following triplet codes for the valine? Okay, so you have to name the codon which codes for valine. So these are the options: GGG, option number B AAG, option number C GAA, option number D is GUG. This is a very straightforward question here. What will be the answer for this question? So let's have a look at the answer. Okay, answer is option number D, GUG. So sickle cell anemia is an autosomal recessive trait. It is caused due to point mutation. What is point mutation? There is a mutation in a single base pair. Clear? So because of this mutation in the single base pair, glutamic acid is substituted by valine at the 6th position of the beta globin chain. Glutamic acid is coded by GAG and valine is coded by GUG. This substitution occurs due to single base pair substitution of the beta globin gene from GAG to GUG. So here you can see here the messenger RNA, you can see GAG codes for glutamic acid and here GUG codes for valine. This is due to single base pair mutation which is occurring in the DNA, which is occurring in the gene. So here initially it was GAG. In sickle cell hemoglobin it becomes GTG. So A is replaced by thymine initially. The next question, person having a genotype IA and IB would show the blood group as AB because of codominance. Okay. This is because of, okay, so what are the options here? Pleiotropy, codominance, segregation, and incomplete dominance. So let's discuss the answer here. Okay, answer is option number B, codominance. So ABO blood grouping system. This is a very classical example of codominance. So what is happening here? You know that the blood group is controlled by a gene I, and this I gene has three alleles, IA, IB, and small I. IA and IB, these are codominant. Both of them are expressed in the heterozygous condition, and the person will be having AB blood group. Next question, ZZZW type of sex determination is seen in. Options are platypus, snails, cockroach, and peacock. So what would be the correct answer here? Okay, let's have a look at the answer now. So here the correct answer will be peacock, option number D. So ZZZW type of sex determination system is observed in birds. So here the females are heterogametic and males are homogametic. Clear? So female consists of two chromosomes, Z and W. So they are producing two kinds of gametes. Half of the eggs will have Z, half of the eggs will have W. Males are homogametic. They have ZZ sex chromosome, so they will be producing only one kind of gamete. All the gametes will be having XZ. In platypus, sex determination system is of XX and XY type. Okay, in snails, the sex determination is environmentally induced. So in some animals, the sex is induced by the environment. Okay, in cockroaches, it is of XX and X0 type. Cockroaches, and in grasshopper also. Okay, XX X0 type of sex determination is observed. Here the females have XX and males have X, only one X chromosome. Clear? Next question, a cross between two tall plants resulted in offering having a few dwarf plant. What would be the genotype of both the parents? So you have to find the genotype of the parent. So you can see the offering. In offerings, the offerings were having few dwarf plant only. Okay, few dwarf plant. Remember this. So dwarf condition is observed when both the recessive alleles are present together. In homozygous recessive condition, dwarfed character means recessive character. Dwarf character, recessive character. So this will be observed in homozygous recessive genotype. Clear? And the offerings, they inherit one allele from father, one allele from the mother. So keeping this in mind, you have to answer this question. So these four options are given to you. What would be the correct answer? So let's have a look at the answer now. Okay, option number B, capital T small t and capital T small t. Okay, dwarfed is recessive trait. Thus, both parents must have at least one recessive allele to produce dwarfed parents. Clear? So both the parents should have at least the dwarfed allele in them, means small t. They should have small t. Only option B and D have small t in each parents. But they are saying only few dwarf parents plants were formed. So option number B will be the correct answer here. Okay. So here you can see only option B and D had small t allele in each of the parents. So you can make the Punnett square for both of them. In both the cases, you can see when the parents have the genotype capital T small t and capital T small t, then you can see you are getting 3:1. Okay. Now, next one. In this case, for capital T small t and small t small t, Okay, here you are getting 2 dwarfed and 2 tall. Okay, although the ratio is 1:1, but here you're getting 2 dwarfed and 2 tall. So answer will be option number B, capital T small t and capital T small t. Okay, clear? So here you are getting only 1 dwarfed. Here you are getting 2 dwarfed. Okay, so for capital T small t and capital small t, number of dwarfed parents is 1, offerings is 1, and for capital T small t parents, dwarf offerings is 2. Okay, when F1 plant they are self-pollinated, then what happens? You get the 3:1 ratio that we have seen. Okay, clear? So this was the correct answer, capital T small t. One of the parent was capital T small t. Another parent was also capital T small t. Clear? So you will get this following generations. Okay, you are getting only one capital. You are getting only one small t small t. So this is 3:1 ratio. Phenotypic ratio here will be 3:1. Okay, so phenotypic ratio is 3:1 here. One dwarfed only. Next question, in dihybrid cross, you get 9:3:3:1 ratio. It denotes what? So when you get 9:3:3:1 ratio in the F2 generation, what does it represent? So this is the phenotypic ratio of a dihybrid cross in the F2 generation. So options are: the alleles of two genes are interacting with each other. It is multigenic inheritance. Okay, or polygenic inheritance. It is caused by multiple allelism. Means when a single gene have more than 2 alleles. And last one, the alleles of 2 genes are segregating independently. So what will be the correct answer here? Okay, so let's have a look at the answer. Okay, so alleles are segregated independently. This is the correct answer. So whenever you get 9:3:3:1 ratio in a dihybrid cross in the F2 generation, the genes are independently assorting. Okay, suppose a cross is made between a pea plant with round and yellow seeds, and this is being crossed with another plant having wrinkled and green seeds. So here 2 characters are taken into consideration: seed shape and seed color. Okay, F1 hybrids are yellow and round because both of these are dominant phenotype. Then you can do the selfing of the F1 generation, you will get the F2 generation. Okay, now in the F2 generation, you will observe the 9:3:3:1 ratio. Okay, so here you can see this is the parental generation. Genotypes is also mentioned. Okay, both of them are homozygous. Okay, both of them are pure breeding lines. Okay, one of the parent is homozygous dominant, another parent is homozygous recessive. You can make the gametes, and then after fertilization, you get the F1 generation. So F1 hybrid will be heterozygous for both the traits. Then you can do the selfing of the F1 generation. Okay, so the F1 hybrid will produce 4 kinds of gametes now. And then you can make Punnett square and you can make all the possibles genotype. And if you calculate the phenotype here, it will be 9:3:3:1. Clear? Okay, what is the outcome of this cross? So this shows that the segregation of seed color is independent of the seed shape, because you are getting new combinations here. You're getting parental combinations and you're getting new combinations also. This is due to law of independent assortment. Okay, here the segregation of one trait, which is seed color, is independent of the segregation of another trait, which is seed shape. Okay, now both the parental and new combinations of the character appear in the F2 generations. These new characters which appear, this is due to law of independent assortment. Therefore, attachment of genes of one pair is independent of the other pair. Now, when the alleles of two genes are interacting with each other, one may dominate over other or become a recessive. Clear? So there was one option that the genes are interacting with each other. So when the genes are interacting with each other, they may dominate over each other or they may be recessive. Now there was one option, multigenic inheritance. So this describes a characteristic that is specified by a combination of multiple genes. So multiple genes are controlling a particular trait. So that's why this is also not the correct answer. Then this is also not example of multiple allelism. So multiple allelism is a type of non-Mendelian inheritance pattern that involves more than just the typical two alleles. Okay, so Mendel studied the traits, contrasting traits. Okay, and each gene had two alleles there. Okay, so Mendel didn't demonstrate multiple allelism. Multiple allelism is a type of non-Mendelian inheritance pattern where one gene has more than two alleles. Next question, this is a very short type answer question. What is the cross between the progeny of F1 and the homozygous recessive parent called? Okay, how is it useful? Okay, so here F1 is being crossed with the homozygous recessive parent. What is this cross called? So let's have a look at the answer now. So this cross is known as test cross. Okay, when the F1 progeny is being crossed by homozygous recessive parent, it is called test cross. Such a cross is useful to determine the genotype of an unknown trait. So why is test cross done? To determine the genotype of the organism which is having the dominant phenotype. Okay, clear? So like suppose you are observing a plant and the plant is tall. So by observing the plant, you cannot tell about the genotype, whether this plant genotype is capital T or capital T small t. Okay, so you can know the genotype of this plant with the help of test cross. Okay, so it helps in determining whether it is heterozygous or homozygous dominant. So this test cross will help you determine whether that tall plant had capital T genotype or capital T small t genotype. Okay, so whether it is homozygous or whether it is heterozygous. Clear? Now, a cross between pure dominant capital A and hybrid dominant capital B individuals with recessive parent is, you can see, shown below here. So basically, you can see if the genotype of the dominant parent is homozygous, if it is capital T, and if it is crossed with the recessive parent, then what happens? Okay, all the F1 generation will be tall. Clear? So if in the F1 generation all the offerings are tall, if you are observing the dominant phenotype, then you can say that the parent is having homozygous dominant. It is homozygous dominant. Okay, it is capital T. But if you are getting 1:1 ratio in the F1 generation, you can say that the genotype of the dominant parent, this is capital T small t. So there are two cases. In one case, you cross this capital T with small t. Another case, you cross capital T small t with small t. Okay, if you're getting all the dominant phenotype, then the genotype is capital T. If you're getting 1:1 ratio, means half tall and half short, means half dominant trait and 50% recessive trait, then the genotype is capital T small t. Okay, now next question. Do you think Mendel's law of inheritance would have been different in the characters that he chose were located on the same chromosomes. Clear? So if the characters that Mendel chose, okay, if these characters were controlled by genes which were located on same chromosome, then will Mendel have got the same results or not? This is the question. So let's have a look at the answer. So yes, Mendel's law of inheritance would have been different in above mentioned scenario. If the genes are located on the same chromosome, then Mendel would have not got the results. Okay, he have not got the 3:1 ratio in the monohybrid cross and 9:3:1 ratio in the dihybrid cross. So if the characters are present on the same chromosome, they would be linked. So linked genes, they are present on the same chromosomes and they are inherited together. That's why you get more parental combinations and you don't get the 9:3:3:1 ratio in the dihybrid crosses or 3:1 in the monohybrid cross. Okay, linked genes would never assort independently. Clear? So Morgan discovered linkage and he was conducting experiments on Drosophila. He conducted dihybrid crosses in Drosophila. And what he observed? He didn't observe the expected 9:3:3:1 ratio. So he concluded that the genes are linked together. That's why he was not getting this ratio. Okay. So this was reflected in the F2 generation where linked genes inherited as a group. So these linked genes are inherited as a group. That's why you get more parental combinations. Next question, you have to enlist the steps of controlled cross-pollination. Would emasculation be needed in a cucurbit plant? You have to give reasons for that. So Mendel conducted artificial cross-pollinations. So you have to list the steps, how the cross-pollination is being done. What is the role of emasculation? Also, they're asking in cucurbit plants. Now, what are the steps of controlled pollination, cross-pollination? First is selection of the parents. So parents should be selected having desired characters. Then emasculation is done. Okay, like the pea plants also, pea plants were bisexual. So these plants were emasculated, means the anthers were removed so as to prevent the self-pollination. We want to prevent self-pollination and we want to cross-pollinate the, you can say, flowers with the desired pollen. Okay, next step is bagging. So the emasculated flowers, they are bagged now. Why they are bagged? Because they are bagged to prevent the pollination from unwanted pollen. Clear? So they are bagged with the help of a butter paper. Okay, so this bagging prevents contamination of fish sigma with unwanted pollen. Then what is done? Now next step is pollination. So the pollen from the desired male parent, okay, pollen from the desired, you can say, male parent having the anthers, they are collected from the anthers and dusted on the stigma of the emasculated flower. Clear? Now next step is the fruit development. So then the normal development takes place. Clear? After pollinating, the flowers are again bagged. Clear? They are rebagged to allow fruit formation. Clear? Now, emasculation is not needed in the cucurbit plant. So they were asking, would emasculation be needed in cucurbit plant? So this is not needed in the cucurbit plant. Emasculation is essential only in the case of bisexual flowers to prevent self-pollination. In the case of cucurbit plant, female parent produces usually unisexual flowers. Okay, so cucurbits have unisexual flowers. So here emasculation is not required. The next question, a person has to perform crosses for the purpose of studying inheritance of few traits or character. What should be the criteria of selecting the organism? So what will be the answer here? Now, the criteria for selecting organisms studying inheritance are, first of all, you should have well-defined characters, means the characters should be distinct and contrasting characters. The flower should be bisexual. Okay. Why bisexual flowers are being used? To allow self-pollination. So what Mendel did, Mendel self-pollinated the F1 generation. Self-pollinating means selfing. So Mendel, okay, he did the self-pollination of the F1 generation, and he did the cross-pollination also of the parents. That's why bisexual flowers were chosen by the Mendel. Okay. Now, short lifespan. Okay. If the plant is having short lifespan, it can produce, you can say, seeds in less number of time. Clear? So lifespan should be short. So you can study the inheritance pattern in short duration of time. Then pollination procedure should be simple. Okay. And that plant that you are selecting, the plant should produce large number of seeds or large number of offerings. Okay. So if there are large number of offerings, then there will be large number of variations that you can study. And if the sample size is large, then your results will be quite robust. Yeah, you can do statistical analysis on large population size. The next question, so here a pedigree chart is given to you. Okay, so a pedigree chart given below shows a particular trait which is absent in the parents but is present in the next generation. Irrespective of the sexes, you need to draw your conclusion on the basis of pedigree. So the pedigree chart is shown to you. Here they are saying the parents, these are the parents, male and the female, both the parents are unaffected, but their offerings are diseased. First of all, this information is given to you. Okay, so you have to draw the conclusion about the inheritance pattern here. And you can see one more information is given to you. This disease is present in the next generation, irrespective of the sexes. So both the male and the female, they are affected equally. Okay, so what will be the answer here? Which type of inheritance pattern is this? So what is the answer here? So you can see no parents are affected. Okay, only 2 of the offerings are affected among 5 offerings. 1 male and 1 female offering is only affected. What are the conclusion then? So we conclude that this trait is showing autosomal recessive inheritance pattern. So you can see what happens in autosomal recessive inheritance pattern. The parents are carriers, so they are not showing any disease, but the offerings are diseased here. Clear? So both the parents should be carriers here. So they are carriers and they are unaffected. And in some of the offerings, you can see some of the offerings are diseased. When they are diseased, when they are inheriting both the copies of the mutated gene from the parents. Okay, so this is the recessive condition which expresses in the homozygous condition. So they should inherit both the recessive allele from the parents. And why this is autosome? This is autosome because both the males and females, they are affected equally. So they are saying irrespective of the sexes. So when they are saying irrespective of the sexes, it is linked to autosomes. Okay. Clear? So here the parents are carrier, they are heterozygous. And because of this, the offerings, okay, as a result, offerings only few show the trait and irrespective of the sex. So that's why it is autosome. Other offerings are either normal or they are carriers. Okay, where capital is for normal allele, small is for the diseased allele. So you can see in the diseased, this is diseased male having small a, diseased female is also having small a. Okay. So the parents are carriers, you can see here. And the offerings, which are diseased, they are inheriting small a from father, small a and small a from mother. Okay. And this disease is irrespective of the gender. Male is also affected, female is also affected. The next question, in order to obtain the F1 generation, Mendel pollinated a pure breeding tall plant with a pure breeding dwarf plant. Now, but for getting the F2 generation, he simply self-pollinated the tall F1 plant. Why? So Mendel cross-pollinated the parental generation, but Mendel self-pollinated the F1 generation. What is the reason for that? So let's have a look at the answer. This is a very good question. Now you know characters segregate during the gamete formation. Okay. Now, pure breeding parents give rise to F1 with heterozygous condition. So Mendel selected pure breeding parents, means they were homozygous for the characters. And these pure breeding parents, when they were crossed, they produced F1, which was an hybrid. Okay, it has heterozygous condition. Clear? And only the dominant trait was expressed there. Recessive trait were not expressed in the F1 generation. Now, only self-pollination of the heterozygous can result in all possible recombinations of characters in the progeny, as mating is random. So heterozygous, the F1 hybrid, which was heterozygous, it was self-pollinated because it will only result in all possible recombinations of the character in the progeny, as mating is random. So Mendel wanted to study, okay, about the different recombinations of the character, and that can only be observed when the F1 generation is self-pollinated. Okay. The next question, genes contain the information that is required to express a particular trait. You have to explain this. So genes control a particular trait. Okay, what will be the answer for this question? Let's have a look at the answer. Now, the genes contain the information required to express a particular trait. This can be explained by the following experiment. So Beadle and Totem set up an experiment to prove that one gene possesses a particular trait and is responsible for the production of one enzyme or the protein. So what Beadle and Totem said? Okay, so they did an experiment which proved that one gene controls a particular trait, and one gene is responsible for the production of only one enzyme or protein. So they performed their experiment on Neurospora crassa, which were nutritionally mutant. Okay, so this Neurospora crassa was nutritionally mutant. Okay, it was proved that a single protein contains several polypeptides, and each polypeptide is controlled by separate genes. Okay, so you know proteins, they are polypeptides. And here each polypeptide in the protein was controlled by separate genes. Okay, thus each gene expresses a particular trait. So one gene codes for a one protein. Okay, what Beadle and Totem showed? So they showed that one gene codes one protein. And this one protein controls one trait. It controls one phenotype. Another gene produces another protein or another polypeptide, and this controls another trait. Third gene, this produces another polypeptide, and this controls another trait. Okay, so this theory was called one gene, one enzyme, or one gene, one polypeptide hypothesis. Means one gene produces only one enzyme, or one gene produces only one polypeptide. And this one enzyme and one polypeptide controls only one trait. Okay, but after discovery of cistron, which is the function unit of gene, the theory was named as one cistron, one polypeptide hypothesis. Okay, so this theory was later modified and it was given the name one cistron, one polypeptide. Okay. Clear? Now you know genes, they are the segment of DNA, and DNA is a polymer of the nucleotides. And DNA contain instruction for building and maintaining an organism. Clear? So DNA consists of all the informations, and genes, they are the segments of the DNA. Now instructions are encoded within a specific sequence of nucleotide in the gene, and this ultimately determines the trait. So basically, you can say that genes which are made up of DNA, these genes are, or these genes which are made up of DNA, this is transcribed into mRNA. mRNA is translated into proteins. And proteins, they are enzymes. Okay, so one gene is coding one protein, and one protein controls a particular trait. The next question, in a Mendelian monohybrid cross, the F2 generation showed identical genotypic and phenotypic ratios. Okay, in F2 generation, you can see phenotypic and genotypic ratio are identical. What does this tell about the nature of alleles involved? You have to justify your answer. Now, identical genotypic and phenotypic ratios in the F2 generation indicate the incomplete dominance of the dominant allele over the recessive allele. So in the case of incomplete dominance, you get the same phenotypic and the genotypic ratio in the monohybrid crosses. Okay, what is incomplete dominance? Where neither of the allele is dominant over the other. Or in other words, you can say in incomplete dominance, what happens? The dominant allele is not completely dominant over the other allele. That's why the other allele is also expressed. Okay. Now, properties of the allele involved in incomplete dominance. So, heterozygous displays an intermediate phenotype here. Okay. So, here the heterozygous will demonstrate intermediate phenotype. Here, the phenotype is a blend of both homozygous phenotypes, or it is a distinct phenotype not seen in either of the homozygous parent. So in the F1 generation, in the heterozygous condition, intermediate phenotype is observed, and this phenotype is different from both the parents. Clear? You get the ratio of 1:2:1. So both phenotypic and genotypic ratio are 1:2:1. Now the heterozygote has a unique phenotype here, which is intermediate phenotype. Okay. And this is different from both the homozygous parent. Clear? And this is different from the complete dominance. Okay, this is incomplete dominance. This is different from the complete dominance, where only dominant trait is observed. In incomplete dominance, heterozygous would look the same. Okay, in case of complete dominance, what happens? The heterozygous looks the same as that of homozygous parent. Okay, but in case of incomplete dominance, you get the intermediate phenotype. Okay, now example of incomplete dominance is flower kernel in snapdragon. Okay, so here the plant having red color flower is crossed with the plant having white color flowers, and the F1 hybrid have pink color flowers. Okay, so this is a blend of red and white color flower. This is due to incomplete dominance. So here you can see parental generations. So there was a plant having red color flower. This was crossed with the plant having white color flowers. In the F1 generation, you get all the pink flowers. And selfing of the F1 generation will give you F2 generation. In F2 generation, you see the phenotypic ratio is 1:2:1. 1 red, 2 pink, and 1 white. And genotypic ratio is also 1:2:1. The next question, can a child have blood group O? If his parent have blood group A and B, you have to explain. Okay, can a parents having blood group A and B, can they have a child having blood group O? So let's have a look at the answer. So yes, a child can have a blood group O if the parents have blood group A and B. Okay, blood group O means that child will have the genotype small I, small I. Okay, so this is only possible when the parents, okay, the parents are heterozygous. Both the parents should have at least one I allele. Okay, then only the offerings, okay, then only the child can have O blood group, because it will inherit one I from father, one small I from the mother. Okay, so this means the father should have a genotype IA and small I, and the mother should have the genotype IB and small I, or vice versa. Clear? So there can be two cases here. Either the mother can have blood group B, IB small I, or the mother can have blood group A. Similarly, father can have blood group A having genotype IA small I, or the father can have blood group B having genotype IB small I. Okay. So basically, both the parents should have at least one allele that should be small I. Now you can make all the possible combination. You can see here, one of the offerings is having blood group O, and the other offerings are having blood group AB, B, and A. Okay, this is clear. Now, next question: what is Down syndrome? And you have to give it symptoms and cause of the Down syndrome. Now, why is it that the chances of having a child with Down syndrome increases with the age of mother? Okay, when the mother exceeds 40 years, then there are the chances of the child to be born with Down syndrome. Why is this so? So let's have a look at the answer. So Down syndrome, it is a genetic disorder. Okay, it is a type of chromosomal disorder. What happens in Down syndrome? There is a trisomy of 21 chromosome. So usually there are two chromosomes. Okay, there are 21 chromosomes. But here there are 3 pairs of 21 chromosomes. That's why this is known as trisomy of the 21 chromosome. Okay, this was described by Langdon Down in 1866. What are the reasons behind Down syndrome? So basically, due to non-distinction of the homologous chromosomes. Okay, non-distinction means failure to separate. Due to non-separation of the homologous chromosome, okay, Down syndrome occurs. So non-distinction of homologous chromosomes, okay, occurs during the gamete formation. So this gives rise to this Down syndrome. Here there is a trisomy of the 21 chromosomes. So this is occurring during gamete formation here. Okay, sometime during gamete formation, what happens? The chromosome do not separate, and one of the chromosome can carry, you can say, 3 copies of a single chromosome. So here there are 3 copies of 21 chromosome in one gamete. Such individuals are aneuploid, means they have 1 extra chromosomes. So they will have 47 chromosomes. Okay, means 2n plus 1. Okay, what are the symptoms of Down syndrome? Mental retardation, growth abnormalities, means short stature with a small round head, furrowed tongue and partially open mouth, dwarfness, palm is broad with characteristic palm crease. And the chances of having a child with Down syndrome increases with the age of the mother. Okay, when the mother is over 40 years, then the chances of Down syndrome increases in the child. Okay, why is this so? Okay, what is the reason behind this? Now, age adversely affects the meiotic chromosome behavior. Older eggs are more prone to non-disjunction. Okay, where the chromosome don't separate properly during gamete formation. So here you can see these are the symptoms of the Down syndrome. This is a child having Down syndrome. All the symptoms are also mentioned here. And this is the karyotype of Down syndrome. Here you can see there is a 3 copy of 21 chromosome. Okay. The next question, how was it concluded that genes are located on the chromosomes? So genes are located on chromosomes was concluded by Sutton and Bovray. So Sutton and Bovray independently in 1902 proposed chromosomal theory of inheritance. What was Sutton's work? So Walter Sutton studied chromosomes and meiosis in the grasshoppers. What are the observations? Chromosome exists in pairs within the cells. Okay. And chromosome pairs separate during the meiosis. Each gamete receives only one chromosome for each original pair. So you can see the chromosomes are behaving very similar to the genes. So you can see like chromosome exists in pair, the alleles also exist in pairs. Chromosomes separate during meiosis, genes also separate during meiosis. And each gamete receives one chromosome, each gamete receives only one allele during gamete formation. So the behavior of chromosome is parallel to behavior of the genes. Why is this so? Because genes are present on the chromosomes. Okay. Now Sutton recognized that chromosome behavior during meiosis mirrored Mendel observation of trait segregation. So basically, chromosomal theory of inheritance tells you that, okay, or it explains the Mendel's law of inheritance with the help of chromosome movement. Okay, so this is how the chromosome move during the cell division. Okay, this is the G1 phase, then you have the G2 phase. Here you can see chromosome duplication has occurred. Then in the meiosis 1 and in the anaphase of meiosis 1, the chromosomes separate, and in the anaphase of meiosis 2, the chromatids separate. In the end, 4 cells are formed. Okay. Now next. Bovray's work. What was Bovray's work? He studied chromosome in sea urchin. What was the observations? He observed his observation aligned with those made by Sutton, suggesting a common pattern. Okay, further he showed that having the correct number of chromosomes necessary for organism to develop normally. Okay, basically both of them gave chromosomal theory of inheritance. So based on these observations, Sutton and Bovary proposed that genes are located on the chromosomes. Okay, now they are saying a plant with red flowers was crossed with another plant with yellow flowers. If their fun showed all the flowers having orange color, you have to explain the inheritance here. So you can see in the F1 generation, you are getting orange flowers. So this means this pattern is incomplete dominance. Neither of the gene is dominant over the other. Okay, you are getting intermediate phenotype in incomplete dominance. So neither of the alleles shows dominance here. Intermediate phenotype is being produced, which is different from both the parents. Okay, clear? So red and yellow colored mixed in the F1 generation to produce orange color. Okay, so this is showing incomplete dominance. Here you can see, okay, red flowers and yellow flowers are being crossed. The genotype is also mentioned. In the F1 generation, R and small r means heterozygous condition exists and orange color flowers are formed. Now, what are the characteristic feature of true breeding lines? Okay, so true breeding lines, they are made by self-pollination. Okay, so it is stable trait inheritance and expression for several generations. So true breeding lines, they show stable trait inheritance. What do you mean by stable trait inheritance? Mean traits, they are passed on stably from one generation to the next generation. The traits, they are maintained in the next generation as well. They are not lost. So the offerings will also have the same traits as that of the parents. Okay, this is stable trait inheritance. So pure breeding lines or true breeding lines, they show stable trait inheritance. Okay, they are produced due to continuous self-pollination. Okay. Now they are homozygous for the trait. Okay, true breeding line, they are homozygous for the trait. Either they will be homozygous dominant or they will be homozygous recessive. For example, capital T or small t. Okay, they show stable trait inheritance, mean trait is passed down from the parent to the offerings over multiple generations. The trait is not lost, it is being passed on from one generation to the next generation. So that's why there is a consistent phenotype. So all the offering will exhibit the same trait as that of their parents. Okay, so you can easily study the inheritance of the trait. Okay, example, a true breeding line of purple flower pea plant will produce all the offering with purple flowers only. Every time you cross two of these plants. Okay, this is the question here, short answer type question. In peas, tallness is dominant over dwarfness and red color is flowers is dominant over white color. Now, when a tall plant bearing red flowers were pollinated with a dwarf plant bearing white flowers, the different phenotypic groups were obtained in the progeny in the numbers mentioned below. Okay, so these were the numbers. You have to mention the genotype of two parents and of the four offspring types. Let's have a look at the answer. So results show that four types of offerings are in the ratio 1:1:1:1. So this is the ratio that you get in the test cross of a dihybrid cross. So in the test cross, so this is a result which is observed in test cross progeny of a dihybrid cross. So this cross can be represented as, okay, so you can see the genotype of the parents are given to you. Clear? One of the parent is heterozygous. Another parent is homozygous recessive. Capital T, capital R, small r, this is heterozygous. And capital small t, small r, this is homozygous recessive parent. Now, the information is already given to you about all the alleles here. Okay, now you can make, using the Punnett square, you can make all the offerings. Okay, so you are getting 1:1:1:1 ratio. Okay, so this is a result of a test cross where one of the parent is heterozygous, another parent is homozygous recessive. Okay, this is clear. Okay, so here the phenotypic ratio is what? 1:1:1:1. Now next question. Okay. So here you can see here. Clear? So this is the next question. In a plant, tallness is dominant over dwarfness and red flower is dominant over white. Starting with the parents, work out a dihybrid cross. What is the standard dihybrid ratio here? And do you think the values would deviate if the two genes in the question are interacting with each other? So here is the solution. Okay. So here you can see the parents are having this genotype. One is homozygous dominant, capital T, capital R. This is homozygous dominant. Another parent is homozygous recessive, having small t, small r. You can make the gametes here. F1 generation will be hybrid here. Then selfing of the F1 will give you F2 generation. Okay. So here you can see you get a typical 9:3:3:1 phenotypic ratio. Okay. And yes, these values will deviate if the two genes are interacting with each other. So they can show linkage or they can show incomplete dominance here. Okay. So you will not get this typical 9:3:1 ratio. Okay. So now due to linkage, the genes will not show independent assortment. So you will not get this 9:3:1 ratio because linked genes, they are inherited together and you get more parental combination. In incomplete dominance, neither of the gene is dominant over the other. Okay, so in incomplete dominance, what happens? You get the intermediate phenotype in the F1 generation. Clear? The next question is a long type answer question. In humans, males are heterogametic, females are homogametic. You have to explain, are there any examples where males are homogametic and females are heterogametic? Okay, so they are asking about opposite of the sex determination system in humans. Also describe as to who determines the sex of an unborn child in humans, and mention whether temperature has a role in sex determination. Okay, let's have a look at the answer now for part A. Okay, we are talking about the answer for part A. So males, they are homogametic. Okay, males are homogametic in case of XY sex determination system. Okay, humans, they have human male, they have XY chromosome, so they are producing two kinds of gametes. Females are homogametic because they have X and X, so they are producing only one kind of gametes. Okay, okay, here. Then humans, they have XX, XY type of sex determination system. Females have a pair of X chromosome, males have one X and one Y chromosome. So females are homogametic here. All the eggs will have only X chromosome. Okay, males will produce two kind of sperms. So males are heterogametic. Okay, half of the sperm will have X chromosome, half of the sperm will have Y chromosome. So this is the sex determination system in humans. Okay. So some examples where males are homogametic and females are heterogametic. So this is sex determination system in birds. So in the birds, females have ZW and males have ZZ. So males are homogametic here and females are heterogametic. Okay. This is clear. So this is the opposite of the XY sex determination system. Where females are producing two kinds of gametes, Z and W, males are producing only one kinds of gametes having only Z allele in them. Okay, Z chromosome in them, you can say. Now, an answer for part B. So, in humans or in XY sex determination system, the male determines the sex of the child because males are heterogametic. Males are producing two kind of sperms here. So, if the sperm having X chromosome, this fuses with the egg, then the zygote will develop into female. If the male having the Y chromosome, okay, if the sperm having the Y chromosome fuses with the egg, the zygote will have XY condition and it will develop into male child. Okay, so there is a 50% chance that there will be a daughter or a son will be born. So there is an equal chance here. Okay. Now as for the next part, role of temperature in sex determination. So temperature can also play a role in sex determination in some animals, for example, crocodiles and turtles. So this phenomenon is known as temperature dependent sex determination. Here the low temperature favors hatching of the female eggs. Okay, and high temperature lead to hatching of the male offspring. So at high temperature, males are formed. At lower temperature, females are formed. Okay, so this is the end of this session. I hope this session was quite knowledgeable for you. Okay, we'll meet you in the next session now. Okay, thank you very much for listening.